Today we're going to take a look at how the electrochemical impulse works across the neuron membrane in respect to this graph, and also we're going to talk about the sodium potassium ions, the channel, diffusion, and the pump, and everything that you see outlined on this page. So first, one setting that we're going to establish is there is a voltage across the neuron membrane, which is similar to potential energy, and the movement of charge equals electrical energy, because we're going to see a lot of movement of these ions, and the movement of charge equals electrical energy. Sometimes you might hear the negative side, the positive side of a neuron membrane, and they say it's negative because if it's more positive on the outside, then it's a little bit more negative on the inside. But they're all positive ions, but they're just, when there's more positive outside, you kind of get a negative here, just to establish a setting. And we're going to jump to the first step, which is polarization, which is just your resting potential or your resting neuron, as this picture has shown us. So what happens is it is around at negative 70 MV, although it might be different, but it's kind of very negative. There is more sodium ions outside, which is more positive outside because there's more sodium outside, more sodium. And what's going on in respect to diffusion and pump is they're both happening simultaneously because it's a little bit leaky. So the diffusion will have potassium moving out, sodium moving in. The pump will be bringing 2 potassium in and 3 sodium out. Okay, so on the graph, it would just be at resting potential. It would be the straight line here. Now the second part is depolarization, which is if a nerve fiber is stimulated strongly enough, it will reach a threshold potential. So if it's stimulated strongly enough, it will reach a threshold potential, which is this area here. If stimulated strongly enough, an action potential will occur. And by action potential, it will be all of this part. The action potential will occur, which might go up to 40 MV depending on your neuron. Sodium gates will open, and the sodium will rush into the axon due to the first one is potential difference, and the second one is due to it moving down the concentration gradient. Okay, so the sodium will all rush into the axon, and what will happen is there will be a very positive charge inside. When it reaches equilibrium, the sodium gates will close. So the next part is repolarization. Once the action potential peaks, and this is the action potential peaking, I'll call it the star, sodium channels will close, the potassium channels will open. So when the potassium channels open, this is the part where it happens. When the potassium channels open, it will make the outside of the axon more positive. It will make the axon outside more positive. So, okay, it will make the outside more positive, except ions will be on the wrong sides of a membrane. I'll show you in a second. And then since the ions are on the wrong side of a membrane, the pump will exchange the ions. So, sodium channels close, and then when potassium channels open, this part will happen, and so the outside of the axon will be more positive because the potassium is going to diffuse outside. It used to be more positive inside from what we got in the last stage in depolarization. The inside was supposed to be more positive, but potassium diffuses out a lot more faster than sodium, so then you'll have a lot of potassium outside, and then that will make the outside of the axon more positive. And now we got that charge, we got the outside more positive, like our resting state, However, the ions are not in the same place. We're supposed to have a sodium ions outside and potassium inside, but in this stage we got sodium inside and potassium outside. So then what we must do is the pump must exchange the ions. So the pump is going to pump sodium outside using ATP. That's energy. And then for a fraction in time, the outside is going to be more positive because the pump is working. Because it's way more positive outside, this is a special part where we call hyperpolarization. Hyperpolarization on the graph is this part. So once you bring the potassium inside, because you don't want it hyperpolarized, you will get back to your resting potential. And potassium getting inside will be using ATP. And I'll show you how the pump and that diffusion process works in a bit. Okay, so then we're going to go back to our normal state, our resting state almost, but it's not exactly in resting state. So you see this part is kind of like the original part back here. Okay, now the last part is a refractory period, which is the time it takes for the action potential to reach resting potential. So it is a time for the action potential, which is a star on the graph, to reach a resting potential, which is the end here. It is a time. It's also called the recovery period because this is a time that must elapse before a neuron can generate another action potential, also called a nerve impulse. So the refractory period is just from here to here because this is a time it has to go through before you can repeat this process from the beginning again. Okay, so I hope that made sense. And I know the pump and diffusion part is a little bit strange, but luckily there is a way to get through it. The pump uses active transport, which requires ATP because it's moving against the concentration gradient. The pump will pump potassium inside and sodium outside, and it does it by pumping 2 potassium in and 3 potassium out. 3 sodium out. As for diffusion, what tends to happen is sodium will diffuse inside and potassium will diffuse outside, and potassium will diffuse faster than sodium. So for example, we see that in depolarization, sodium has been diffusing inside, and then in repolarization we've had this part where potassium is diffusing outside. So in diffusion, that's how it works, and then for the pump you can actually just look back and you'll see that it works like this. Something important is the pump and diffusion during resting potential, so that's the first part. During resting potential is working at the same time due to the leaky pump. Okay, now just 2 more vocabulary that we're going to define for the sake of understanding how the pump works. So the electrochemical impulse runs on an all or none response. So the nerve and muscle cells respond maximally or not at all, or the signal in fiber is all or none. So they're either going to, if they meet a threshold potential, if they just meet this part, they are going to respond to this part. They're going to respond maximally at the star. If they don't reach this part, if they just go here, then they're not going to respond at all. The threshold level means the minimum level of a stimulus required to initiate an action potential. So the minimum amount of stimulus, which is maybe the stimulus goes here or here, but for here, the minimum level of a stimulus to initiate the action potential, this is the action potential, you must at least have this much stimulus to be able to reach here. Otherwise, If your stimulus does not reach a threshold potential, say it reaches here, your stimulus, it is not going to initiate the action potential. So this is a minimum that you must get to get here.